So our today's topic is the process of the tooth development. The process of the tooth development, it begins during the embryonic life, and it involves a series of interaction between different cell layers and the tissue. So to start our discussion on this subject, first of all we shall discuss the initial stages of the tooth formation. During the early embryonic development, the oral ectoderm, it forms the lining of what will eventually form and become the oral cavity. So this ectoderm, it is the outermost germ layer of the embryo. Now there is contact between the oral ectoderm and the endoderm of the foregut. So the oral ectoderm that is forming the primitive oral cavity, it comes in contact with the endoderm of the foregut. So this occurs due to the presence of this buccopharyngeal membrane. So it is a temporary structure separating the primitive oral cavity from the foregut. At approximately 27th day of the gestation, the buccopharyngeal membrane it would rupture. So this event creates a connection between the primitive oral cavity and the foregut that will allow the communication. So below the oral ectoderm, there are specialized cells that are derived from the neural crus cells that are also known as ectomesenchyme. They begin to form the neural crus cells. They are a group of cells that migrate during the embryonic development, and they contribute to various tissues and structures in the body, including those in the head and face. So during the early stages of embryo development, after the rupture of this buccopharyngeal membrane, when embryo is about 6 weeks old, there is an increase in the proliferation of subtle areas of the basal cells within the oral ectoderm. So this rapid division leads to creation of what is known as a primary epithelial band. This band of epithelium extends into the underlying actin mesenchyme along with the future dental arches resembling a horseshoe appearance. So this primary epithelial band, it divides into vestibular lamina. This one is the vestibular lamina and this one is the dental lamina. So by the 7th week, the primary epithelial bands, it divides into two distinct processes. The inner process is known as, it is responsible for the foundation for the future teeth, that is known as the dental lamina. And the outer process, it contributes to formation of space between the teeth and inner lining of the cheek and lips, that is known as the vestibular lamina. So the dental lamina, it serves as precursor for the ectodermal part of the deciduous teeth. Additionally, during the jaw development, the distal, the permanent molars, they will originate from the directly from the part of the dental lamina that is known as the distal extension. So remember, the initiation of first permanent molar, it begins around 4th month in utero. The second molar, it begins after birth. The third molar, that is the wisdom teeth, it starts its formation around 4th to 5th year after birth. So the distal proliferation of dental lamina, it plays an important role in determining whether the germ of these permanent molars will be situated, particularly in the ramus of mandible and the tuberosity of the maxilla. The successor of the deciduous teeth, which are the permanent teeth, they develop from the lingual extension, from the at the free end of the dental lamina, opposite to the enamel organ of each deciduous teeth. So this extension is known as successional lamina, that begins its development from around 5th month in the utero. So that contributes to development of the permanent teeth that range from the permanent central incisors to the second premolars, whereas the vestibular lamina, it would give rise to the oral vestibule. Now this dental lamina, it is a specialized epithelial structures that form an extension or thickening of primary epithelial band. So these thickening along the dental lamina, localized thickening occurs at various points. These thickening will give rise to individual tooth bud that makes the location where the tooth formation. So the tooth bud that forms along the dental lamina, it will correspond to different type of teeth, incisors, canine, premolars, molars, that will develop in the future. Remember the vestibular lamina. This vestibular lamina would give rise to formation of the oral vestibule, that is space between the teeth and the inner lining of cheek and the jaws. What is the fate of the dental lamina? The dental lamina, it is active for the shorter duration. Soon after the tooth development starts, the dental lamina starts to degenerate. So in the third molar region, this dental lamina will be active till the age of 5 years. The dental lamina, it remains functional for shorter duration. So after the initiating the tooth development, the dental lamina, it begins to degenerate. So after the disintegration, the remnants of the dental lamina, it persists as epithelial pearls or island within the jawbone and even within the gum tissue. So these remnants, they are termed as a cell rest of. So certain points in the future, developing maxilla and mandible, the ectoderm bulges and grows into the underlying mesenchyme. There, the 10 tooth buds in the maxilla and 10 in the mandible, they are arranged in the form of horseshoe shape. So each growth of dental lamina would represent the enamel organ of tooth bud of the deciduous teeth. What are the genes that are expressed during the tooth development? Remember, LHX6, LHX7 gene, they are expressed in oral ectomesenchyme by the day 9th of gestation. They originate from the signals in the oral epithelium of the first branchial arc. So these genes are important for the teeth development. Other is the signals from the oral epithelium, including FGF8. It determines the location of the tooth germ. Remember PAX9 gene. It helps to define the precise site where the tooth germ will appear. Its expression matches these locations and it is influenced by this FGF8, and it is repressed by BMP2 and BMP4. So several other genes expressed in the oral epithelium regulates or influenced by these signaling proteins would determine the tooth germ position. Genes like DLX1, DLX2, SHH, SBFA1 gene, PAX, PAX9 gene, they are important for various stages of the tooth development. So defect in the mutation in the gene like remember PITX2, SHH and PAX9 gene, they are linked to conditions that are known as missing teeth. That is tooth agenesis. So this question was asked in your one of your AIMS SRship exam. PAX9 gene, it is responsible for, it causes, they are linked to condition that is known as missing teeth. Now the tooth shape determination, the process of determining the specific tooth type and their correct position, that is known as patterning of the dentition. So for the tooth shape determination, there were two theories that were given. That is, one is the field model and other is a clone model. So according to the field model, it suggested that the distinct factors responsible for tooth shape are within different areas of the oral tissue, overlapping in graded field for each tooth family, whereas the clone model states that each tooth type arises from specific group of cells within the oral tissue that are programmed to produce a particular, that are programmed to give a particular tooth shape. Other is the homeobox code model. According to that, homeobox genes like MSX1, MSX2, DLX1 and DLX2 and BARX1 gene, they are involved in the dental pattern. So the early expression of these genes in the oral tissue defines broader areas where the different type of teeth would develop. So the misexpression of BARX1 gene in certain area of the oral tissue that results in a transformation of incisors to molars. So this question was also asked in your AIMS exam, SRship exam. The misexpression of BARX1 gene would cause transformation of incisors into the molars. So highlighting the influence of these genes on the tooth shape. Now let us break down the process of tooth development and stages into simpler terms. The first is the formation of the enamel organs. Along the dental lamina, which represents the location for the deciduous teeth, there is a rapid multiplication of cells that forms the small knobs. So these knobs grow into the underlying tissue and mark the beginning of enamel organ for each tooth bud. So remember, all the enamel organ, not all the enamel organs start developing at the same time. Usually the ones in the front lower jaw. The enamel organ, it will grow in size and change its shapes as the cell multiplication continues. It takes shape of cap with one side facing the mouth and other side having a concave shape. Eventually there will be formation of dental papilla and dental follicle. Inside the enamel organ, the cells multiply to form denser tissue that is beginning of dental papilla. Surrounding the enamel organ and dental papilla is the dental sac that is made up of cells and fibers. Now let us discuss about the various stages of the tooth development. The first will be the bud stage. The bud stage, it is the initial phase of tooth development and represents the very beginning of the tooth development. At this stage, the enamel organ, it takes a shape of small bud. So within this enamel organ, the outer part it contains the low columnar cells. So the outer part, it is made up of, the peripheral part is made up of columnar cells, and toward the center there are the polygonal cells. So in periphery there are the columnar cells, and in the center there are the polyhedral cells. So the mesenchymal cells that surrounds the developing enamel organ, it begins to proliferate. So they multiply rapidly. As a result of that proliferation, these cells condense or comes closer into two specific areas. The first is the dental papilla, and other is the dental sac. So the dental papilla, it will give rise to dentine and pulp. So you can remember this mnemonic by D and P. So dentine, D stands for dentine and P stands for the pulp. The mesenchymal cells that surrounds both the enamel organ and dental papilla, they condense to form a dental sac. So the dental sac will develop into supporting tissues surrounding the teeth, such as the dental sac would give rise to cementum, periodontal ligament, and the alveolar bone. So remember this for your entrance exam. The dental papilla would give rise to dentine and pulp, whereas dental sac would give rise to cementum, periodontal ligament, and the alveolar bone. So this stage represents the very early stage of the tooth formation, where the enamel organ, it begins to take shape, and the surrounding cells undergo changes, setting the foundation for the structure that will constitute the future tooth. The next stage is the cap stage. So during the cap stage, the tooth bud does not expand uniformly. Uneven growth in the different part of tooth bud result in the creation of shallow invagination or depression on the deep surface. That is what we call it as cap stage. So there will be outer enamel epithelium and the inner enamel epithelium. So this outer enamel epithelium, the cells at the periphery of the cap, they are cuboidal and covers the cap rounded outer surface. So they form the outer enamel epithelium. The cells in the center depression, they become so tall and columnar. So they will constitute the inner enamel epithelium. So at a delicate basement membrane will separate the outer enamel epithelium from the inner enamel epithelium. So this membrane, it is connected to cells by hemidesmosomes that will hold them to the basal lemma. So within the enamel organ, there are polygonal cells in the center. that forms a cellular network that are known as the stellate reticulum. So the stellate reticulum, it consists of polygonal cells located at the center of epithelial enamel organ between the outer and inner enamel epithelium. So uh, due to presence of glycoaminoglycan in a ground substance, the water, it is drawn into the enamel organ from the surrounding dental papilla through osmotic forces. So as a result of that, the polygonal cells, they would become star shaped, and they will remain connected to each other through this their cytoplasmic extension. So this network of star shaped cells, it is known as stellate reticulum. So what is the function of this stellate uh, reticulum? Number one, it it gives a cushion-like consistency and act as a shock absorber. So supporting and safeguarding the delicate cells that are involved in the enamel formation is its function. Additionally, the dense packed cells in the center of enamel organ, they form enamel knot. That projects slightly toward the dental papilla. So the enamel, what is the function of this enamel knot? You can get MCQ. The enamel knot, it plays a role in signaling and determining the tooth shape, but it disappears before the enamel formation. So enamel knot would act as a reservoir of the dividing cells from growing enamel organ. So enamel knot, it plays an important role in determining the shape of the teeth. And it disappears before the enamel formation begins. So the enamel organ, during its development, it forms specialized structure. What are those structures? They are the enamel knot and enamel cord. Enamel knot, the cell at the center of the enamel organ, they become densely packed. They are known as enamel knot. And when the enamel knot, it gives a vertical extension, it is known as enamel cord. So enamel cord is vertical extension of the enamel knot. So when the enamel cord meets the outer enamel epithelium, when the enamel knot, it meets the outer enamel epithelium, it is known as enamel septum. So this structure would divides the stellate reticulum into two sections. At the point where the enamel cord meets the outer enamel epithelium, a small depression would occur that is known as enamel navel, and it resembles as similar as umbilicus. Functionally, the enamel knot and cord, they were initially believed to be act as a reservoir for the dividing cells for the growing enamel organ. But recent studies, they have revealed that the enamel knot, they act as a signaling centers. There will be formation of the dental papilla. The inner enamel epithelium influence the surrounding actin mesenchyme, causing it to proliferate and condense. So this condensation would result in formation of dental papilla that will give rise to dentine and pulp. Simultaneously, at the enamel organ and dental papilla, there is a denser and more fibrous layer that is known as a dental follicle.
The next stage is the bell stage. The bell stage, it consists of following layers in the enamel organ: outer enamel epithelium, inner enamel epithelium, stratum intermedium, and stellate reticulum. In bell stage, the enamel organ, which is like a little pouch in the mouth, it takes a bell-like shape. So it was once thought that the shape of the tooth crown was because of the pressure from the cells in the growing dental papilla. So what opposes this pressure from below? Well, the fluid that is present in the stellate reticulum, it pushes back, balancing the pressure that is from the below. And how do we get different shape of tooth like pointy parts or cusp? It is because of how the cells divide and change. The cells that are present in the future tip of tooth stop dividing earlier and starts to change first. So as they change, the other cells around them keep pushing them. These early changes, uh, so this early changing the cells to stick out, making a cusp or point on the tooth. So the shape of the crown, it is decided in this stage. So each tooth shape comes from these early changing cells, and between these points or cusp, the parts of tooth slope down gradually because of cells dividing and changing from tip to the base. So the tooth shape that we call as tooth morphogenesis, it is controlled by special instructions from the genes and signaling molecules. So these are like the bosses that gives order to shape the tooth. So we can see the following layers in the microscope: the inner enamel epithelium, the stratum intermedium, stellate reticulum, and outer enamel epithelium. The inner enamel epithelium, which is a single layer of cells that turns into ameloblast. So they are like tall, skinny cells that are attached to each other by lateral junction, and they are connected to cells in the stratum intermedium through desmosomes. So these ameloblasts play a big role in the formation of enamel. So are those ameloblasts the ones that make the tooth enamel? Exactly. They are the one responsible for making enamel. Now uh, in between the inner enamel epithelium and stellate reticulum, there is an intermediate layer that is known as a stratum intermedium. So this layer is important for the enamel formation. It has a squamous cells which are closely packed and that seems to be very active metabolically. Moving on to the stellate reticulum, it is made up of star-shaped cells with long processes that connects to the neighboring cells. So the stellate reticulum, it expands due to increase in the fluid, but when the enamel formation begins, it collapses, bringing ameloblast closer to the nutrient capillaries. So why is that important? So this collapse will ensure that the ameloblast, which are the enamel making cells, get all the nutrients they need for the intense activity. Now the outer enamel epithelium that is present on the outside of enamel organ, It starts as a cuboidal. The outer enamel epithelium will start as a cuboidal cells but later flattens out. So before the enamel forms, this layer will fold, creating space for nearby mesenchymes to form papilla that brings in lot of nutrients for enamel making process. Lastly, the dental lamina, it extends and forms what we call the successional dental lamina. So this is important because it leads to development of the enamel organ for the permanent teeth. The dental papilla, that is the part of innermost part of the enamel organ, it starts to transfer into odontoblast. The cells that makes the dentine. And just before the dentine starts performing, there is a special membrane that is known as membrana preformativa that separates the enamel organ from the dental papilla. Other important structure is the dental sac. So dental sac, it is an important structure that surrounds the developing tooth because the formation of dental tissues. So as the tooth root starts developing, something important happens to fibers within the dental sac. They change and they develop into the periodontal ligament fibers. So periodontal ligament fibers, they are the special fibers that get embedded into the cementum and the alveolar bone. So yes, the dental sac, it transforms into these periodontal fibers. The next comes the advanced bell stage. The advanced bell stage marks the beginning of the mineralization and root formation in the teeth. So at this stage, something important happens. The boundary between the inner enamel epithelium, which eventually becomes the meloblast, and odontoblast outlines what we call the future dentino-enamel junction. So what happened next? Well, the first dentine formation begins at the future dentino-enamel junction, primarily in the area where the future cusp will be located. Then the meloblast, which will have differentiated from the inner enamel epithelium, start laying down enamel over this dentine in the region that will become incisal and cuspal area of the tooth. So the root development also started at this stage? Yes, absolutely. The cervical part of the enamel organ, it contributes to formation of Hartwig epithelial root sheath. So that play an important role in so this Hartwig epithelial root sheath, they play a role in shaping the root and initiating the radicular dentine formation. Hartwig root epithelium sheet, remember, it is formed by the outer enamel epithelium and the inner enamel epithelium. So as the root development progresses, the Hartwig epithelial root sheet, it loses its structural continuity and forms a network of cells that are known as epithelial rests of Malassac. So that is what is important for your entrance exam point of view. So these reft of Melasses, they are found in periodontal ligament of the ruptate tooth. So does this Hartwig epithelial root sheet. It plays a role in multi-rooted teeth. Yes, indeed. In teeth with the multiple roots, the differential growth of Hartwig epithelial root sheet, it caused division of the root trunk into 2 or 3 roots. So the expansion of the enamel cervical opening, it results in tongue-like extension of the horizontal diaphragm. So these extensions, they grow, fuse and divide into cervical opening into 2 or 3 opening, leading to formation of the multiple roots. So what happens when there is a disruption in this Hartwig epithelial root formation or continuity? So if there is any disruption, it can lead to certain issues like enamel pearls defects in the dentinal wall that could cause the development of the accessory root canals. So that will summarize the complex process of the Hartwig epithelial root sheet and the root formation in the teeth.